So we're there.
Good to go. Have at it. Thank you.
Hallelujah.
Thanks everyone for your patience.
Yeah, don't we just love it when you have to update the Zoom app and that causes problems. Okay, so it looks like we're good. All right, so Allison, are we good to go?
Yes, we are good to go.
Okay. All right, well, it's a pleasure to be with everyone, and my name is Greg Nordin, and I'm very pleased to be joining you from, well, actually, there we go, from the BYU campus here in lovely Provo, Utah, right below the mountains. And what I'd like to do today in terms of our topic is let's first introduce it in the following way. Imagine that you were to have been born 200 years ago, so during the time of your fifth, sixth, seventh great grandparents, and imagine what your life would have been like at that time, and then consider what it's like today. And I think you'd agree that the world that we live in today is truly shaped by technology. And what we can do with our technology or in our technology is really defined by the tools that we have and the tools that we develop. And so if we want new possibilities, we have to create new tools. And so the topic I'd like to discuss with you today is lab on a chip technology, which is a new tool to enable taking a biomedical laboratory, for example, and shrinking it down to a little tiny chip that's maybe a centimeter on a size. And so this is something that could be relevant in the following way. Many of us have had to go get blood drawn, always a favorite experience. And then once that blood is drawn, then it goes to a central laboratory where there are these big machines, and then whatever biochemical analysis is going to be done then happens in that machine. And so you need a fair amount of blood in order to do those analyses, and you need expensive reagents and whatnot. And so that comprises then whatever test is being done. On the other hand, some of us have had the experience of getting a finger prick and donating much smaller amount of blood, and then having that used for whatever the biomedical test is. And so that's kind of the idea with microfluidics, is taking a much smaller sample volume and introducing it to a little tiny chip that deals with that small fluid volume plus much smaller volumes of whatever reagents are required for the biomedical assay that is being done to learn whatever needs to be learned from that blood sample. And so this is called lab on a chip technology, and has been around for a couple of decades, or I should say in development for several decades. A lot of exciting things have happened, but there have been some hangups with it. And what we're going to address today is one way that we can overcome some of those difficulties. And just a word on terminology, lab on a chip is often also called microfluidics. And so we're going to talk about 3D printing for microfluidics and what that enables and how that overcomes many of the problems that traditional fabrication technologies encounter. So for example, when you try to make a microfluidic device, the key thing that you have to be able to do is create these interconnected hollow regions in a bulk material. So it's all about those hollowed regions, or we call them negative features or voids, because that's where the fluid and the reagents are going to be. And so how those hollow regions are arranged and what's in there, that really comprises the functionality of that microfluidic device. And when we say microfluidics, we really mean micro voids, so things on the order of 100 microns or less. So let's get a sense for the size scale that we're talking about. Over on the left, we've got a little kernel of brown rice. Looking at our scale bar down here in millimeters, that's maybe, I don't know, 2.5, 3 millimeters wide and 4 or 5 millimeters tall. So we're all pretty familiar with that. If you have a poppy seed muffin, that poppy seed is about 1 millimeter in diameter. Or if you take one of the hairs off your head and look at the diameter of that, that's about 0.1 millimeters, which is 100 microns. So we're talking then about negative features or hollow regions where the dimensions are down smaller than the diameter of that human hair. And the way that that's traditionally accomplished with with clean room and other types of fabrication technologies is to create a series of individual layers, and in the layers create some surface relief features and perhaps some through holes, and then take those layers and align and bond them. And now we have a series of interconnected hollow regions, and we have our microfluidic device. And you notice through this fabrication method, it's a stacked two-dimensional configuration. So mostly you're working in 2 dimensions, and the number of layers that you can bond together is pretty limited. A handful is probably how I would turn it. If you only need to do 2 or 3, life's great. If you need to do 6, 7, 8, oh, it's going to be a very tough life. And so, so you don't get many layers out of this. And like I mentioned, typically you need a clean room to create the masks or to create the molds or other processes, depending upon what materials you're using. And there are a variety of different plastics that are popular in elastomeric materials. So that's how traditional microfabrication is done. And what I'd like to introduce you to is how we now fabricate microfluidic devices in our labs. We used to do that traditional approach. We've done that for many years. And I had enough frustrations with it that about 10 years ago decided that there had to be a better way. And so we started looking into alternatives, and we'll get to that. So as an example device, just to kind of illustrate the process with, let's consider this little 3D printed device that one of my students is holding that's intended to investigate the process of cell chemotaxis. Now, chemotaxis is the motion of cells in response to a chemical stimulus. And typically that chemical stimulus is in the form of a concentration gradient. And so if it's a chemoattractant concentration gradient, the cell will want to go to the higher concentration regions in that gradient. And if it's chemorepellent, then it will be the opposite. And this chemotactic behavior of cells is fundamental to life and to many different types of disease states, as well as to embryonic growth, et cetera. And so it's a set of biological processes that are extremely important, some of which are understood, but some of which are not. And in order to elucidate those processes, then it'd be very useful to have chips that allow us to create very specific and controlled chemical gradients in order to probe this particular aspect of biology. Now, in this chip, then the concentration gradient region is this little tiny channel shown in blue here. So we want to seed cells in there and set up a concentration gradient across that little tiny channel. And the way we do it is we've created 2 reservoirs that we put our source and sink materials in for, excuse me, the concentration gradient that we want to set up. Then we have 2 tiny little 3D printed pumps that pump fluid from the appropriate reservoir into these large channels along the side. That allows us to get an advection-free, diffusively generated concentration gradient here. And then we have the fluid just flowing out to waste. And so that's the chip that we want to fabricate. Here's what it looks like after fabrication. And then if we characterize what's going on in this little concentration gradient region with some fluorescent material, we do see a change in fluorescence as we go from the source fluid down to the sink fluid. And so this operates as we would hope and expect and as it's designed to do. So now we perform a chemotaxis experiment or a series of experiments. And so here we've got some cells that are seated in this concentration gradient region. We have high concentration and low concentration, so we get the gradient across there. And then there are different ways that we can analyze this, but just in the interest of time, let's consider something called a forward migration index. The mathematical details aren't really that important, but basically positive numbers just mean cells are going in one direction versus another direction. Here we have the experiment and a control where we have no concentration gradient. And you look at these 2 distribution of results and they're clearly different. And in this particular case, the cells are moving in one direction, namely they're going in the direction of the concentration gradient. So bottom line is that the device works, the assay works, we're seeing the clear effects of cell chemotaxis. But now let's talk about fabricating that chip in the first place. So because we found early on that commercial 3D printers did not have the resolution or other features that we needed in order to 3D print microfluidic devices, which remember are characterized by these interconnected hollow regions, then what we found is that we just had to build our own 3D printers that had the capabilities that we needed. And so here we've got a CAD drawing of one of our 3D printer designs that we use a lot. If we take away a lot of the mechanical components and just focus on the main elements, we have this optical engine that projects an ultraviolet image, reflects off this mirror, goes through a window in the bottom of this resin tray, and then that series of images are used to form layer by layer whatever device we want on the underside of this build platform. And so we're projecting, excuse me, images with 7.6 micron pixels. So this is very high resolution in the UV at 365 nanometers. And so if we look at a fabrication run, starting with the software that we developed, this is the initial interface. Then if we start a job, and so we're going to print one of these devices. Here we start a stopwatch so we can time it. You can see that the build platform here is going up and down. Every time it goes up and down once, that's one layer being fabricated. Each layer in this particular case is 10 microns thick. Looking underneath, you can see a little bit of a bluish thing here whenever the UV engine turns on to project an image. Here's the tilt mirror, and then this is the interface. We're monitoring the forces of what's going on in real time so we can do some control and analysis things. And then when we're all done, comes up out of the resin tray. And so it's taken us just a little over 11 minutes to print this device. And so we've tuned the process to not only be very high resolution, but also to be very fast in comparison with other 3D printer approaches. Now, excuse me, it turns out that we have generalized the 3D printing process because we found that typical 3D printing is just too restrictive for the types of things that we want to do. So we published a paper several years ago in Nature Communications that details how we actually do 3D printing. And the long and short of it is, if we look in cross-section at some microfluidic feature we're trying to print, let's say this is just a simple channel, so there's nothing there, and then all this other stuff is polymerized resin. Then we have these layers, let's say these are 10 micron thick layers, and so we polymerize them one after another until we get to this channel region, and then we decide that we need to have different layer thickness here, different exposure times, therefore different states of polymerization, and that all has to be embedded in these regular 10 micron layers that everything else is built out of. And then above, we decide we want some other layer thicknesses and exposure times for various reasons. And so basically, our point is that you can mix and match layer thicknesses and exposure times arbitrarily or nearly arbitrarily throughout the 3D print. And that gives you a lot of additional flexibility in terms of the types of structures that you can fabricate, which allows a very high degree of sophistication to be achieved for these microfluidic devices that I'll show you in just a few minutes. So it turns out no one else does this, though. And so this has been puzzling to us and annoying to a certain degree. We'd really like other people to have these kinds of capabilities. So one thing that we've done in that direction is to open source a 3D printer file format, a specification format that basically encodes all these different capabilities and processes. So anyone can go to GitHub and pull the specification down and create 3D printing technology based upon it. Now, all of that is well and good, but really the key thing is, can you make very tiny negative space features that are in the size domain that are needed for these microfluidic devices? And prior to this publication in 2017 that we put out, really that had never been demonstrated. The best that people had done were hundreds of microns in size, which is, fine for a certain class of applications, but for the things that we were interested in, it was just way too big. And so that was why we ended up developing all of these new 3D printer technologies and techniques. And so this was the first paper then that really highlighted those capabilities. And this scanning electron microscope image really showed the field that, oh, 3D printing can be made a player in this field and a really nice way to do fabrication. And so here we show a channel that's 20 microns wide and 18 microns tall. And this was so much better than anything out there. This created quite a stir and really, I think, opened people's minds to the possibilities of 3D printing for truly microfluidic device fabrication. And so that was 2017. Where are we at now in 2023? Well, we're now down to where we can do channels that are 2 microns wide and 1.9 microns tall. In other words, we've reduced the linear dimension by a factor of 10 in each dimension, so the cross-sectional area is reduced by a factor of 100. So we've been able to make tremendous progress in continuing to push the resolution of what we can fabricate. Now, we'll talk a little bit more about that in a few minutes, but first, let's look at the kinds of things that we can do just based upon the little bit lower resolution stuff, which is already very high resolution that I just showed with this sort of approach. And so first, let's talk about active devices. In particular, the most basic active device is a valve. And if you can 3D print valves as part of your 3D print, then you can do a lot of fluidic control right on the chip itself, which dramatically reduces the amount of expensive reagents you need, et cetera. And so in the upper left is a CAD drawing of the basic design of what we call a membrane valve. If you cut it apart, this cylindrical region here is bisected by a very thin membrane that's simply a part of the 3D print. So it gets 3D printed with everything else. It's made out of the same material as everything else. And then the point is that when you pressurize the upper region, that deflects this membrane down and closes off access between these two channels. And therefore, this valve is in a closed state. When you release the pressure, then the elastic strain on the membrane pops it right back up. And now you have fluidic access between the channels, and now the valve is open. So by applying a pneumatic pressure, then you can open and close these valves on chip. And so if we take a look at these microscope photographs, in the upper row here, we're looking at a side view image of one of these valves in the open state and in the closed state. And here you can see this very thin transparent region, that is the membrane. And over here, when it's actuated, it's closed, then you can't see it because it's lying flat on the bottom of this chamber. But looking at the top view, then you can see that this valve body is basically circular in diameter, and it's 150 microns in diameter. So it's quite small, a little bit bigger than a human hair cross section. And we use 20 pixels in the projected UV image to create the diameter of that thing. And we found that we can push the size even smaller. This shows a 6-pixel diameter valve, in other words, 46 microns. Here in the open state, you can see this red dyed fluid is going through it. And now in the closed state, that membrane has collapsed to the bottom, closed off fluidic access, and now the valve is closed. And so looking at it from the top, 6 pixels is not very much, so it's not exactly round per se, but nonetheless, it works quite effectively. Now, once you have valves, then you can make pumps. So in the upper left, we show an inlet valve and an outlet valve sandwiching what we call a displacement chamber, because its function is only to displace fluid volumes. It never actually closes. So that means we've taken the center channel for this particular structure and moved it over to the side, so it never closes. It just pushes fluid around. And looking at a microscope image from the side, you can see that the fluid fills the regions that should quite nicely. You can see the 3 membranes very clearly, and top view similarly. Then if we go and characterize what kind of flow rates we can get out of this, here we're looking at measured flow rate in microliters per minute. And that's a unit probably most people aren't familiar with, but a microliter, if you do a finger prick to get some blood out of your finger, or to do one of these little finger prick assays, the size of that droplet, or the volume of the droplet, is roughly, let's say, 30 microliters. So this is microliters per minute. And so that's not a huge flow rate, but for these little tiny devices, that is plenty. And so it works well. So if we look at a 50 millisecond phase time here, so we have 5 phases that we or 5 states that we actuate this thing in, and that causes pumping. We'll see that in just a minute. If the phase time is 50 milliseconds, then the flow rate is 0.1 microliters per minute. So if we look at a video of the pump in operation, you can see that as we actuate these membranes, sure enough, we have fluid flow in this vertical serpentine channel, and it works great. I mean, there's no problem. If for whatever reason we need faster fluid flow, one thing we can do is just change the size of this displacement chamber so that during each cycle, we push more fluid out. So here we've doubled the diameter, therefore we've got 4 times the volume. And when we watch it flow, then of course it's flowing faster for the same phase time. So bottom line is you can make pumps, you can 3D print these pumps, they're very effective, they work great. We also invented a different type of valve called a squeeze valve, where we have a channel that we narrow here and cause it to go between a split pneumatic or control channel. So if we look in cross section, here's our fluid channel, and then here's our split control channel. And right here, top and bottom, there are little membranes. And again, this is just all 3D printed out of the same material, such that if we pressurize the channels here, it basically collapses the membranes and closes off the fluid channel. So now the valve is in a closed state. Releasing that pressure, the membranes pop back up, and now the valve is in open state. So again, looking at the top row, here is looking from the side. Here is the valve in an open state. Here's the valve in a closed state. Looking from the top, again, open state, closed state. Here you can see the red fluid has been squished out. And the remarkable thing here is that the active area is 4 by 4 pixels in terms of the projected ultraviolet image. And so that's a 30 micron square region. So this is a really tiny valve. Now, interestingly, we can make it even smaller. We can reduce the overlap area to 2 by 2 pixels. So the active valve region is only 15 microns square, which is really quite remarkable that we can get it to be that small. And yet using this generalized 3D printing approach that I mentioned, that's really what enables us to do this, plus having full control over all aspects of our 3D printer functionality, and we also design our resins to work well with this. Now, once you have a squeeze valve, of course, you can make a squeeze pump. So here you have that narrowed channel going through 3 control pneumatic split channels. And so looking at it from the side, we can see the structure quite clearly, also from the top. And then if we look at flow rate as a function of phase time and look at 50 millisecond phase time, that's giving us 8 nanoliters per minute flow rate. To put that in perspective, that's the same flow rate you have from 8 human sweat glands. You know, when you're outside and it's hot and you're really sweating, that's 8 sweat glands worth of fluid flow out of this pump. So very low flow rates, but nonetheless for a variety of different applications, you know, exactly what one would need. And so here we see a video of the squeeze pump in operation. And yes, we have fluid flow. It's quite effective. And yes, it's slow, but it's got to be slow because it's so small. In fact, speaking of small, you know, here's 100 microns for the scale bar, diameter of human hair. And notice that the pump is only a little bit bigger than that. So this is just really a tiny, tiny pump. And so our 3D printing approach is effective in creating such structures. Now, another longstanding issue with microfluidics is anytime you want to mix things, because of the nature of the physical regime the fluid flow is in these tiny structures, there is no turbulence. Instead, we have what's called laminar flow. And that's a problem when you want to mix things, because turbulence is hugely your friend when you want to do mixing. So to solve this problem, what we've done is we 3D print these very tall, narrow, high aspect ratio channels. So you see this one is 30 microns wide. And then we flow 2 sheets of fluid we want to mix side by side into this narrow region. And now we can use the physical process of diffusion to rapidly cause mixing. And it's rapid because the distance we have to diffuse over is only half the width, or 15 microns. So if we look at diffusion time as a function of diffusion length for some different molecular species, we won't talk about the details. Bottom line is we need to diffuse 15 microns, And that's about in the several hundred millisecond range in terms of diffusion time. So if we can have our fluids dwell in the high aspect ratio channel for a fraction of a second, then diffusion will take care of doing all the mixing for us. Now, when we take one of these high aspect ratio diffusion mixing channels, we can put it together with several valves. And so looking at it from the top, well, valves and pumps, looking at it from the top, we have a pump here for fluid A and a pump here for fluid B. So we can pump in these narrow sheets of fluid into this tall diffusion channel and let diffusion take place. And this shows a microscope image of exactly that structure after fabrication with a blue and red dyed liquid going through and being pumped into this diffusion channel. And when we quantitatively characterize what's going on, we don't have time to go through the details of it, but when you do the characterization, yes, we do get a 50-50 mixture, and it works just extremely well and also very fast. Now, once you have a single one-to-one mixer like that with its associated pumps, you can take 10 of them and serially connect them so that the output of one feeds into the input stage of the next. And then you can take two fluids, fluid A and fluid B. Fluid B would be the diluent, and mix them in 10 stages and doing a twofold or one-to-one dilution between each stage. So this is a 10-stage serial dilution device. So the output fluids here should cover 10 factors of 2 concentration. And if you look at the actual fabricated or 3D printed device, this is what it looks like, and it forms just extremely well. And then you measure the concentration as a function of the output stage, starting with an input concentration of 100%. And what you find on this log scale is that over the 10 output stages, you do indeed get a linear decrease on a log scale of the concentration covering 3 orders of magnitude, which is really great to have that scale of dilution in a very tiny volume on chip. So that would allow you, for example, to do an on-chip assay, dose response assay, just in a single shot. If you look at concentration as a function of time, it takes less than 50 seconds to reach steady state on those outputs. And so if we go to a video of the device in operation with just some black fluid here just to illustrate, and jump ahead in the video to about 50 seconds, it's now in steady state, and all of these outputs are as one would expect. And so that works quite well. Now, all of that was done with the basic technology of membrane valves. We could instead take a squeeze valve approach, and so have a very tiny diffusion channel. Now it doesn't have to be so tall and narrow because it's the flow rate is smaller, but we have 2 side-by-side pumps here and an output valve. So looking at it from the top, fluid A comes in the top, fluid B in the bottom. Here in the microscope image, the green and red fluids mix quite well in the diffusion channel and then are going out into the serpentine. And so here we have a video of this thing in operation, and it works, as one would expect. And the key thing here, though, to note is look at this scale bar. I mean, this is 1 millimeter. So this is 2.2 millimeters wide, 1.1 millimeter tall. This thing, the entire 10-stage twofold serial diluter, is much smaller than a single grain of rice. And this is all 3D printed. And so that's really quite amazing that you can get down to these size scales with 3D printing. Now, another example or two, something else that's quite useful is porous membranes, particularly when you're doing cell work and organoid work and organ on a chip type of work. And so here we've developed a way of doing pores as small as 7 microns in thin membranes. And so we have collaborators that we work with to do a bunch of cell studies with this. Another thing that we needed to figure out several years ago is to be able to connect one chip to another chip and have a very high density chip-to-chip interconnect technology. There are reasons why that's important, but we don't really have time to go through them. The point, though, is that by taking a vertical channel and simply as part of the 3D printing a little tiny gasket around it, we can make a leak-tight seal that seals to quite high pressures. This shows a 1.5 by 1.5 millimeter area in which we have 121 such interconnects. So that's 53 interconnects per square millimeter. So it's a very effective and high density interconnect technology for chip-to-chip applications. So one of my students, just for fun, he decided to create very tiny chips that use this chip-to-chip interconnect technology. And then on each chip, he's just making what for us is a gigantic pump. And so this chip is 2.1 millimeters wide, 1 millimeter tall. And then basically there are 5 chip-to-chip interconnects here so that this pump can be interfaced to another chip. So here with some tweezers and fingers, that sort of sets the scale for one of these chips. And he can print 117 of these in a single shot. So on our current 3D printers, you know, we could do this print in maybe 5 minutes. And so 117 chips every 5 minutes for continuous operation. Of course, there's setup and teardown time too in there. But this shows the pump chip then connected to a very simple interface chip that my student used for testing. And the bottom line is that it works quite well. Okay, so let's go back to the very tiny channels that we now do. We don't have time to go through how we do this yet. In fact, we're preparing a publication that will go through that. But with that capability, we can make little tiny things like this biocage. This is less than 0.5 millimeter in diameter. And if you look at all these little pores, they're very tiny channels. So you can do things with little tiny organoid types of or spheroid types of tissue agglomerations for a variety of applications. You can also go at the mixing problem a little bit different. This is a test chip that we printed just as in a preliminary look at doing a different kind of mixer. So here we're zooming in and looking at this. If we look at it from the side, we've got 2 channels coming up from underneath and then going over the top of each other. If we look from the top, each of those channels splits into 6 mini channels. Those are all interleaved with one another. And then those go into this, again, mixing, narrow mixing channel. And the bottom line is that we can reduce the mixing time from several hundred milliseconds down to about 1 millisecond. And so this gives a lot of flexibility in being able to rapidly mix fluids for on-chip bioanalysis. And this shows an example with some fluorescent dye in the upper channel. So it has some brightness to it. This is totally dark because there's no fluorescent material in it. And then you get this interleaved fluorescent, bright and dark fluids that then mix. And now in this region, they're well mixed. Okay, so we're about out of time here. So let me just summarize. Then our focus has been developing 3D printing for microfluidics. And we found that we've had to create our own 3D printers and materials in order to really be effective and get down to the size scales we need for these negative space features. And so we have this generalized 3D printing approach, and we've tuned things so that our fabrication times are very short. So we've got fast fabrication. I've shown you how we can do different types of valves, membrane valves, squeeze valves. I've given you an example of how you can get a high degree of component integration with the example of the 10-stage 2-fold serial dilution device. Multi-resolution is how we actually get to kind of the next level of the very tiny things that I showed you. And there are some things that we need to do, and you know, there are things that you need in order to accomplish what I've showed. One is the optical properties of the resin have to be tailored correctly, and then you have to have high resolution projection optics. And then it's really critical to have full control of all 3D printer functions. And so that's really what drove us to create this 3D printer technology. So we have more printer development that we're doing. We spun a company out to commercialize our previous developments. There's lots of room for more research in various areas, in particular doing 3-dimensional structures. And then the beautiful thing with this whole approach is you can take a fail fast and fail often development tack in order to explore the parameter space very rapidly and get to a device that works. And then ultimately, of course, we want to get to automated design. So I'd just like to thank my collaborators, other faculty here at BYU, and my wonderful students, graduate and undergraduate students, and our various funding sources. And with that, thank you very much. It has been a pleasure.
Thank you, Dr. Nordin. We will close at this point, and this will be on our YouTube channel. So if you want to come back to it, you are welcome to do that. We should have it within the next couple of days cleaned up and on our YouTube channel. So thank you everyone for joining us, and we'll look forward to seeing you next month.